Yeah. What? Okay. All right, ladies. So I'm gonna add to the basic stuff in genetics. We've talked about some really basic genetics, and today what we're gonna do is we're gonna talk about maybe some slightly more complicated scenarios. So we've made an assumption so far, because we're starting with the basics. We made an assumption that when we're looking at a characteristic, like color or length of fur, that it's one gene that determines that characteristic. And the gene comes in two versions, two alleles. So for color, it would be, in this case, black or brown. For length, it would be long or short. The other assumption that we made, and I haven't told you about this, some of you may have noticed there was a term. The term, where's the term? Right here. Never went over this with you guys. Autosomal. The other assumption we made is that the gene that we are looking at, okay, is on the autosomal chromosome. Now, what exactly does that mean? If you remember the picture of genes, so if you remember, if we go back to a karyotype, not here. All right, let's see, where's this? Genetics. We looked at, there it is, right here, sorry. Okay, if you recall this picture here, okay, picture of your chromosomes, right? Which pair determines gender? It's the sex chromosome, so it's this pair over here, right? So these other pairs, they're called the autosomal chromosomes. This last pair, sex chromosomes. So everything that we've been looking at so far, we've assumed that it was a gene on an autosomal chromosome. That's significant because that means there's no difference between men and women when it comes to the inheritance pattern. All right? So we made an assumption that we're talking about a characteristic that is determined by one gene, that the gene has two alleles, two versions, that one version is dominant over the other. These are the assumptions that we make to start with the basics. And lastly, that the gene is on an autosomal chromosome, which means that there's no difference between men and women because men and women both have two sets of autosomal chromosomes, one from mom and one from dad. So guess what we're doing today? Today we're going to talk about some more complicated scenarios. Things where we can't really make those assumptions anymore. Now what I'm going to show you is not really that more complicated. It's pretty simple. Same logic that we're going to use. So let's talk about the first one. Let's talk about, okay, what if you have a gene on a sex chromosome? Okay, how is it different? Because the difference is this. You have two copies of an X chromosome. Men only have how many? One. So what if you have a gene on an X chromosome? Well, you guys have two copies, men have one. How do we determine the inheritance pattern? So let's look at an example. Let's look at color blindness. Okay? Now, when I give you an example, don't make the assumption there's only this scenario that could happen. So like, there are three genes for seeing color. One of the genes is on chromosome number 4, which means that women have two copies, men have two copies. That's not sex-linked. The other two copies are sex-linked because they sit on the X chromosome. So over here, we see a chromosome. We see the X chromosome. And on the X chromosome, we have a gene that allows us to see a particular color. Most likely it's green. This is probably why it's colored green here. So on the X chromosome, there's a gene for green and there's also a gene for red. we will talk more about the green and red opsin gene that allows us to see color when we get to the unit on evolution, because it has an interesting story. So the difference is this. Women have 2 copies of it. So if a woman is heterozygous and the little c stands for a defective copy, which means that you produce proteins that cannot see green color, and it's recessive, then this female should still be able to see green because she has 1 dominant copy. And remember, for you to see a recessive phenotype, you need to have what? 2 copies. But here's the difference though. Not when it's sex-linked for males. Because if you're a male and you only have 1 X chromosome and it happens to carry the defective allele, then guess what? You have the condition. Because there is no other allele to hide it. So that's something that we've discussed before. If you have a recessive allele, but you also have a dominant allele, you need to have 2 copies of the recessive to see it, like cystic fibrosis, Tay-Sachs, right? But not here. If you're a male and you have a recessive allele on your X chromosome, you show it. So, but how do you determine like the odds of children getting it? Well, it's the same thing. Let's take a look at this cross. Imagine we have a carrier female, okay, she's heterozygous, and she has a child with a colorblind male. How do you do the Punnett square? Well, if you look at the way you do it, it's the same. There's no different. You have a male can produce 2 types of sperm cells. One with the X chromosome, right? And so you can see a male can produce a sperm cell with either this one or this one, and a female can produce only X chromosomes in her egg cells, right? So if you look at the Punnett square, what do you see here? Well, here's my female. She's heterozygous. So one of her X chromosomes could carry the normal C, the big C, and the other one could carry the recessive C. Same idea though. She was what? She was X big C, X little c. So, one possibility or the other possibility. No different. Yeah.
If we put the C like at the bottom of the square, are you going to mark it wrong?
I don't care. It doesn't matter to me. We just put the X there to remind us that the gene is where? Not on a regular chromosome, but on the sex chromosome. Because you got to remember that. Otherwise, you might forget. Now, if the male is a colorblind male, the sperm cell will either have the X or the Y. So does this Punnett square look any different? No, it's still a 2 by 2 square. And how you interpret it is still the same. So if this egg cell meets this sperm cell, what do you get? Well, you get one copy of the normal C and one defective copy. You have two X's. What is this? This is a female. And she's what? Carrier. But can she see green? Yeah, she can. The only way a female can't see green is if what? Yeah, you get one defective copy from mom and one from dad. So in this case, that will be a colorblind female. So there's no way, if you're a female and you have a condition on your X chromosome and it's recessive, the only way you can get it is if your dad has it. He has to show it. Because where do you get your other X chromosome from? Your dad. Now if you look at the male, so male only needs one copy. So here, in this case, male gets a normal X, or male gets the defective X. You notice there's no such thing as a carrier male here for this condition. Because a carrier is what? Heterozygous. And a male can't be heterozygous because they can only have one allele. They only have one X chromosome. Either they have it or they don't. Okay? So, this is called sex-linked. All right, it's the way you figure it out is still the same, still a probability, right? Still Punnett square, all the same, but it's the way you interpret it is different. Okay, so that's one slightly more complicated thing. Let's take a look at another one. Oh, actually, let's talk a little bit more about this. This is interesting. So you guys, we said that you have 2 X chromosomes, right? And male only have 1. So here's a problem. If you have a gene that sits on the X chromosome, you would expect, because women have 2 copies of that gene. Remember I said to you, like, think of a gene like a recipe, right? So if you have 2 copies of the recipe, you might predict that you might make twice as many things from that recipe. If you have 1 copy, you would expect maybe that individual makes less. Something happens though with you guys. As you develop, one of the 2 X chromosomes actually shuts off. It becomes what's called a bar body. So one of the 2, you can see it over here, actually becomes turned off. Okay? And which one it is random. It's totally random. So in one part of your body, you could very well have 1 X chromosome that's disabled, and then another part of your body could be the other X chromosome that is turned off. Now, this is important because well, first of all, it explains something really interesting that we could see in females. There's a gene that determines the ability to produce sweat glands, and it's on your X chromosome. So if you're heterozygous and you have a defective copy, well, if you're a male and you have a defective copy, you can't make sweat glands. But if you're a female and you're heterozygous, what you will find is that in the area of your body where the X that had the defective copy was switched off and the normal copy was left on, you have sweat glands in that area. But in another part of your body where the defective copy was left on and the normal copy was switched off, you have no sweat glands. So you could have a female that has sweat glands in some parts of her body and missing sweat glands in other parts of her body. Well, then you gotta be careful. You don't want to overheat yourself, right? So I'm going to show you another example of this. This is a calico cat, and you'll notice something about the cat. The cat is orange and black. Now, but the white coloration is due to something totally different. So I'm only going to talk about the orange and black. And the idea is this. The idea is this is only possible in females, and you're going to tell me why it's only possible in females. So this cat was heterozygous. The gene for black and orange are sitting on the X chromosome. So she had 1 copy for orange. So I'm going to do this. I'm going to say X, orange, X, black. Okay? So it's heterozygous. So how do you explain the black and orange pattern? It's very simple. One of the 2 gets switched off, right? So as this cat's developing, in the region where the X black is switched off and the X orange is left on, what color will that cat, in that area, what color will it be? It'll be orange. So like over here, we can imagine that the cells in this area, the black was switched off and the orange is left on because one of the X's gets turned off. In the area where we see black, it would be the opposite. The X orange copy was switched off and the X black was left on. So my question to you is, can you get this pattern with males? Why not, Lauren? Right, they only have 1 X. So you can't have this double coloration in males, okay? So, sex-linked, it's a little bit trickier, but as long as you understand the rules and how to interpret what's going on, males only have 1 copy, females have 2. There should be no problem in figuring out how to solve a sex-linked problem. Yep. Yeah. No, that's normal. That's what happens in mammals. Now, yeah, the reason is something called dosage compensation. So that if you have 2 copies of a gene and males only have 1 copy, you can make twice as much of something compared to males. But by switching one of the 2 off, for that protein, males and females make the same amount. For some reason, it seems to matter that males and females make the same amount of these proteins. Yeah, Pam. What color was it? Who? No, it's calico cats are orange and black. That's a different thing. That's determined by a different set of genes. No, it's the whole cat. Part of it is black, part of it's orange, except for the white underbelly. I don't know. I would have been black and orange.
I thought that happened as it developed.
Right. So this happens pretty early in development, like at around maybe somewhere like 10 to a few hundred cells. As the embryo is still really tiny, then you can think of an embryo like a ball of cells, and in one particular, like the North Pole, this copy would be switched off, in the West, this copy would be switched off. So this happens really early on in development. Okay. Okay, so let's look at some other examples of things that are not so straightforward. We assume that a gene determines a characteristic, and we assume there's 2 copies, and we assume that 1 copy is dominant and the other one is what? Recessive. Well, guess what? Might not be the case. So in your lab where you're making the baby, and you have questions about inheritance patterns, so your inheritance patterns are going to be either autosomal dominant or autosomal recessive. So what would be an example of an autosomal recessive inheritance pattern from your list? Chin shape. Which one? Is it the round chin? Is it recessive? I think so.
Because the 2 little R's is...
Okay, yeah. So if you have
the 2 little R's is square.
The 2 little R's is square?
Yeah.
So if the 2 little R's is square and your child is square, has a square chin, then that's autosomal recessive. If they have round and that's dominant, then you would say it's autosomal dominant. If the 2 conditions blend, what would be an example of blending in your baby lab? Like straight hair with curly hair gives you what? Wavy. If your child has wavy hair, then that's blending. Okay. So blending is known also as incomplete dominance. So, what color is this flower? What color is this flower?
White.
And so this is pink. Now, how do you figure out? Well, if I have a white flower cross a red flower, I get pink flowers. If I take 2 pink flowers and I get them to have babies, how do you figure out? It's the same thing. What do you notice? It's still a Punnett square. It's no different than what we did before. So, what do? Well, a pink flower is heterozygous, right? So it's R1, R2. R1 means red, R2 means white. If we cross 2 pink flowers together, the way you interpret it is just different, but the way you set it up is still the same. This individual can only produce R1 and R2, right? You're either going to get an R1 or an R2. This individual over here is either going to give you an R1 or an R2. So the way you set it up is still the same. So this would be R1, R1. But what does R1 stand for? Well, R1 is red. So what do you get here? Red, red gives you red. What do you get over here? Well, this box is R2. with R2. R2 is white. So R2, white is white. Now here, R1, R2, because you got R1 with R2. Well, it's red with white. If red was dominant over white, then this would be what? This would be red. But it's not. So if it's incomplete dominance, it's blended. Right, they both show up. So they're pink. Now I'm going to show you codominance. Codominance is a little bit different than blending. Okay? So in blending, I like to think of it as like the average out. The average out, right? Codominance, they both show up. So an example is, imagine that if this was codominance, this flower over here would not be pink. It would actually be red and white. So it would be like maybe red with some white in it, or white with some red in it. Maybe. Maybe. Now I'm making that up, but that's what codominance means. Codominance means that you see, they don't blend, you see both of them. So I'm going to show you an example, okay? So, blood type. Now, blood type, we know that there's different types of ways you can organize blood type. One is through the ABO system. So, let me show you. Can you see that? I'll use it. So, let's imagine that you have blood type A. What exactly does that mean? Well, that means that in your genetics, you produce a type of protein that I'm going to represent as a square. And if you have blood type B, you produce a different type of protein that I'm going to draw as a triangle. If you have blood type O, you produce nothing. You don't produce the square or the triangle. They don't exist. Now, if you're blood type AB, you produce the triangles and the square. You don't produce some object that's in between a triangle and a square. You produce both. So if you see both, what is that? That is codominance. Alright, that's codominance. So that's an example of codominance, whereas blending would be incomplete dominance. So here are the blood types. Now, I don't really care if you use I, or you just leave it as A, B, and O, but I'll show you an example using this. Oh, actually, one little thing. A and B are codominant, but O is recessive. Okay, O is recessive to A and B, but A and B are codominant with regards to each other. So let me show you an example. Okay? I'm going to just come up with a question. All right, so we're going to make up a question. Yeah, go ahead. Yeah.
So in the column, like, would you write like either incomplete or no?
Yeah.
Yeah?
Yeah.
You'd either write incomplete dominance or codominance.
Yeah. But we don't have any examples of codominance. It'd be incomplete or complete dominance or complete recessive. Or there's one more, which we'll talk about in a second. This is faster. I'm using red for blood type, get it?
I found out what my blood type was when I was 15%.
Oh yeah?
Neither of my parents.
What is it? What's your mom? What's your dad?
I got the polycystic from my mom and my dad.
So let's do that.
But my best friend's mother has B positive blood type, so I'm staying in contact with her.
So here's for here. Yeah.
That's so mean.
Is it Francesca with a K?
No, it's a C.
Leave it with a K. What's your blood type?
I'm B positive. And mom is A positive.
All right. Now the positive and negative have to do with something called a rhesus factor, and it's one of those things where it's different than the A, B, and O. Okay, it's different. Positive means you have it. Negative means you don't have the factor. This one is dominant. This is recessive. So if you're positive, that means you have at least one copy of the dominant rhesus factor. Let's just talk about the B, okay? So you're saying you're B.
Yeah.
And your mom is A. And your dad is AB. So how did you get to become B? So listen, your dad is AB, right? So if you think about a Punnett square, your dad can pass on A or B. Your mom is A, but guess what she has also with her? She has the O. Well, think about it. Because
oh my gosh.
this gives you A. This gives you AB. Now, because your mom also has the O allele, you could be blood type A, or you could have BO. Blood type B. Now, how do I know if she has the O? Because let's think about if she didn't.
Does that mean I have the O? I'm just B?
Yeah.
So I'm no.
Yes.
That's such a crappy blood.
So let me explain why your mom cannot be AA. Because if she was AA, then if you did the Punnett square, you're either going to be A or you're going to be AB.
Or she just dropped it. How do you know her mom has an O?
We just proved it. We just proved it.
If my mom had AA, I could be B.
Yeah, if her mom had AA, then that means A's are possible and AB is possible, but no blood type B by itself. Yep. Yeah, so let's imagine we had a cross between a person who has blood type A, right, with someone who has blood type B, but they're heterozygous, so they're AO and BO. So these are your possibilities. This is codominance, AB. This is just B, because B is dominant over O, because O is recessive. This is A, and this is O. So the only way you get all four possibilities is if you have AO cross BO. So with the blood type, yeah, O is recessive to AB, but AB are codominant with respect to each other. Okay? So the way it works is pretty simple. Your immune system learns, and what happens is, after about a year or two, your immune system has learned that if you're blood type A, then anything with a square is normal. If you're blood type B, anything with a triangle is normal. So that's why you got to be careful. You can't just get any blood. If you're blood type B and your immune system says triangles are normal, squares are not, and you get blood type A, guess what's going to happen? You're going to reject it. Okay. Now the reason why your immune system cannot reject blood type O, you can't reject what you can't see. Okay. But you'll learn more about that in grade 12.
Oh.
All right. So, the other thing we've considered that we kind of, so I'm going to skip over some stuff. We've going back to a dihybrid cross. You remember what a dihybrid cross is?
That's two.
Right. And we assume that they don't have anything to do with each other, right? We treat them separately. Well, guess what epistasis refers to? They do affect each other. So sometimes one gene can affect another one. And if it does, We call it epistasis. So, let me show you an example with Labrador retrievers.
I love
Right?
retrievers.
So, this is 3 different labs. These are the codes. Sorry. It's a little bit bigger. Okay. What's the only way you could be a yellow lab?
If you have, I don't know.
According to the code here.
Oh.
2 little e. So, what's going on here? There's 2 genes for color. The B stands for a gene that gives you black or brown. Now in this case, black is dominant over brown because black, as you can see, is got the capital B. We scroll down a little bit. You can see the capital B for black, whereas the brown is little b. But there's another gene called E. And that gene, E, determines whether or not you're even going to get the black or brown. So if you have a capital E, it says, okay, you can have color, and then the color is either going to be black or brown. But if you have little e's, it says no color, which means it doesn't matter what the other ones are, because you have 2 little e's. So in this case, the E does matter. The E does affect the letter B. When one gene affects another one, we call it epistasis. Okay? So here's another example of that. Here, one gene, to make a color called anthocyanin. Over here, we need 2 genes. So, what would happen if we have a cross between this? Big C, little c, big P, little p, big C, little c, big P, little p. Normally we would just treat them as a dihybrid cross, right? We would work it out. So we need to get this color. What do we need? We need 1 big C and we need 1 big P. That's what we need. So what would happen if you had 2 little c's? No color. What would happen if you have 2 little p's? No color. So what's the odds of getting no color with this cross? Well, here's the thing. How do we do this question? That's physics. You don't want to see that one.
I got myself.
Right here. So let's take a look. So in that example, we have big C, big P, little p, cross, big C, big P, little p. And we need big C and big P for color, because they affect each other. So how do you do this question? Well, same way you did your other one. It's still a dihybrid cross. So what do? We set up a Punnett square. It's a horrible Punnett square, I agree.
Why does it just do one line?
This is better for me.
No, it's not. It looks like a wave.
It is a wave. So here. It is horrible. Alright, so what are the possibilities? Well, what can this individual give you? They can give you big C with big P. So big C, big P. Or big C with little p. Right? Big C with little p. Or little c with, or, okay. And this individual over here, same, right? So, how do you do the Punnett square? Does it look any different? No, it's exactly the same. Big C, big P, big P. Color, yes or no?
Yes.
Yes. What about this one? Big C, big P, little p. Yes. What about this? Big C, big P, big P. How about this one? Big C, big P, little p. So all of these are color. So far, 4 color. Now, this one. Big C, big P, little p. Alright, that's 5. Big C, little p, No. No color. No color. Alright. Little c here, big C here. So big C, big P, little p. Alright, 6. Big C, little p, No. So we have so far 6 color, 2 no color. Look, do this row over here. Big C, big P. That's another one with color. So now we're up to 7. Big C, big P. Now we're up to 8. Little c, big P. No. So now we're up to 3. So now, so far, we have 8 with color and 3 with no color. What about this one? Little c. So now we have, so far we have 8 color, 4 no color. But look, is it any different? Like, did we set this up any differently? No. But how do you interpret the answer? Well, it's a little bit different because one gene affects the other, right? So big C, big P, little p. What is that? Color. All right. What about these ones? Big C, little p. No color. Little c. No color. That's 6. And this one, little c.
No color.
7. So we have 9 with color, 7 without color. Again, same setup, same logic. You still do the law of independent assortment. You still separate the C's and P's, but then the way you interpret the results is different. Okay? All right. Let's stop there. That was long. All right?
37 minutes long.
Okay?
Bye.
The end.